Hello everyone, welcome to my channel. Today I'll be going through part 1 of module 2 chapter 2 of the OCR biology A-level specification, which is the basic components of living systems. I'll specifically be going over microscopy, so from 2.1 to 2.3 of the A-level biology textbook, and part 2 will go over 2.4 to 2.6. This is focused for OCR, but it can be useful for other exam boards. Please subscribe to help my channel grow, and I'll continue to make these videos covering the A-level biology specification. Every organism is made of one or more cells, which means that they are either unicellular or multicellular. Microscopes allow you to magnify organisms in order to see details into them that aren't fully visible to the naked eye, things like cells and their organelles. So, from GCSE, you probably already know about light microscopes and electron microscopes, but for A-level biology, you'll need to know the types of electron microscopes, which are transmission electron microscopes and scanning electron microscopes. Firstly, a light microscope is the type of microscope that you've probably encountered at school. The components that you should know of this are the light source, stage, objective lens, the coarse and fine focus adjustments, and the eyepiece lens. So the way light microscopes work is that when a specimen is placed onto the stage after it's been prepared and the microscope is on, the light source should shine through the specimen, through the objective lens, and into the eyepiece lens, which allows you to see a magnified image that can be adjusted to different magnifications. Although, with a light microscope, a limiting factor is the resolution because you can only see so much. Then there are the two types of electron microscopes, transmission electron microscopes and scanning electron microscopes. Transmission electron microscopes work by a beam of electrons transmitting through the specimen to produce an image, magnifying it onto a layer or a screen that allows it to be seen. Scanning electron microscopes have a beam of electrons going across the surface of the specimen, reflecting to produce an image that's 3D, magnifying it and allowing it to be seen. For TEMs, the resolution can be about 0.5 nanometers, and for SEMs, it can be between 3 to 10 nanometers, or around that. In other words, they can be 0.0005 micrometers, or 0.003 to 0.01 micrometers. Sometimes, when using electron microscopes, scientists can run into these things called artifacts, which are small imperfections on a microscope that can be mistaken for details on a cell. It used to be more common in the past for scientists to mistake these for actual organelles, but now we have methods to distinguish them and differentiate them so that it doesn't cause any errors. So here's an overview of similarities and differences between the different types of microscopes that we spoke about. Although you don't really need to know the values, you should know light microscopes have the lowest resolution, scanning electron microscopes have higher resolution, and transmission electron microscopes have the highest resolution. Again, you don't really need to know the values, but light microscopes have the lowest magnification, while transmission electron microscopes and scanning electron microscopes have higher magnifications. Light microscopes are more cheaper and affordable than both kinds of electron microscopes, and light microscopes are more portable than both kinds of electron microscopes. Light microscopes use light and so they form coloured images, but TEMs and SEMs only produce black and white images. Both light microscopes and transmission electron microscopes produce 2D images, but scanning electron microscopes produce 3D images. The specimens for light microscopes can be dead or alive, but for TEMs and SEMs, since the electrons are passed through a vacuum, the specimens must be dead. In all cases, the specimens should be very thin to be seen. On the screen, you can see an example of the images that can be produced by each microscope. You can see that the light microscope has a coloured image that's 2D. The transmission electron microscope produces an image that is also 2D, but is black and white. The scanning electron microscope produced a 3D image that is also black and white. I've been using the words magnification and resolution, but what do they actually mean? So magnification is how many times larger an object is in an observed image in comparison to its real size. To remember the equation for magnification, just remember I am. I image size, A actual size, M magnification. And when you rearrange it, you get magnification equals image size over actual size. In real life and an exam, the actual size and image size will have different units, and so you need to remember to convert between them. And now, the resolution. Resolution is the smallest distance in which two objects can be distinguished from one another. For example, if something has a resolution of 2 mm, you might look at it every 2 mm and see it as two different objects. But the moment you zoom in to get into 1 mm, they are no longer two distinguished objects and merge into one. Some people do find this part more complicated, which I understand, so I'll try to make it simple. For light microscopy, differential staining is very important in order to differentiate and identify organelles and improve image contrast. It's not super necessary that you learn the name of each of the stains that you can use, but there have been questions in the past that ask you to suggest stain names. So I'm going to name some and you can take them down if you want. Acetic aursine makes chromosomes appear dark red. Methylene blue makes DNA appear blue. Eosin makes cytoplasms appear pink. Iodine makes starch appear as violet or blue. Sudan black makes lipids appear black. You can prepare the specimen itself in a few different ways. For example, a dry mount is where the specimen is cut into thin slices or viewed as a whole, and a cover slip is placed over the sample to keep it from moving or being moved. A wet mount involves the specimen being dropped with a liquid over it and a cover slip placed at an angle over the sample. This allows living aquatic organisms to be viewed. The reason why it's done at an angle is to not allow air bubbles to form, because doing this pushes air to one side. Squash slides can be done with this method. The specimen is dropped with a liquid and a cover slip is pressed down gently onto the specimen and the sample can be squashed. Smear slides are where the sample is smeared with the edge of a slip to create a thin layer and then a cover slip is placed over the sample. This can be done with things like blood samples. For your specification, it's very important that you know how to produce a scientific drawing for these images. I'm going to tell you what you really should and shouldn't do when producing a scientific diagram drawing because it's very important and you can lose marks if you get it wrong. Always remember to add a title and a scale. This is very easy to forget and overlook, but it's really important if you want to get full marks on the question. In your drawing, make sure the lines are clear and continuous. You shouldn't sketch and you should not shade in. Your drawing should be large and fill over half of the page, and the proportions should make sense. When you label, make sure the lines you use are straight and typically at one side, and they have to be done with a ruler. Your labels shouldn't have any arrowheads and they should not cross over in any way. If you follow these instructions, then it should be very easy to get full marks in a question asking you to draw. Here I'm showing you an example of a type of drawing that wouldn't get many marks, if any at all. As you can see, the lines are not very clear and continuous. There's quite a bit of sketching, and although there's a title for this image, there is no scale or magnification. As for the labels, the arrows do have arrowheads and they overlap, and also some of the labels are just wrong. This drawing would very likely not get many marks, if any at all. So that concludes part 1 of chapter 2. Later on, I will post a part 2 as well as a video on how to calibrate a microscope, which is very important for your PAGs and exam. If this was helpful, please leave a like and comment on what I can improve or what I should go over next. Good luck!